Right, here we go. Are we already live?
Yes.
Great then. Hello, everybody. Hello, everybody. Good afternoon. I'm really happy that you are all with me here. And you can salute everybody who's listening and see us through YouTube of the channel of the Safe AgroBee project. Well, it is an honor for me that all of you accepted to be part of this meeting and celebration, because it's a kind of a joint meeting of four PRIMA projects that they all do care about bees. And I'm also very, very honored that apart from people working for these projects, coordinators or partners, doesn't really matter, we are all equal. We have with us the officers of PRIMA Secretariat, that they are responsible for these projects, and it's really good for them to take the time to be with us for this celebration. Thank you so much about that. And also we have with us the representative of FAO, Rosita. Also, thank you, Rosita, for your time. And somebody representing Apimondia, who is the vice president. It's Peter Cosmos. And with us also is the dean of Apimondia, Zil. So, welcome everybody, and thank you again. I have nothing else to say than we should enjoy this day, because this way we will have a good understanding of what's happening with all these four projects now. Maybe there are even more, but we don't know about them. And let's start from the welcoming words. Yeah, maybe I would like the colleagues from PRIMA Secretariat first to welcome this event. So who's going to start? Sarah, maybe?
Okay. Hello. Thank you so much for having me today. I am Sarah, and I am the communication officer at PRIMA. And today also we have Eda, who is also representing PRIMA and the PRIMA Secretariat and the project officers. Well, it's a real honor that you have invited us to attend this lovely celebration. It's also very interesting to see that there are some synergies and collaborations happening between different projects of PRIMA, so that no one is working alone, and whenever we are stronger when we work together and when we collaborate in events like the ones that you have prepared today. So it's a real honor having us. Just to give a short introduction, as you may know that the UN has designated the May 20th as the World Bee Day. The main goal of this day is basically to raise awareness of the importance of pollinators and the challenges that they face. And this day shows that the pollination is a fundamental process for the survival of the ecosystem. And not only do they contribute directly to the food security, but also are crucial for the biodiversity. The FAO this year has celebrated the World Bee Day, and they have organized an event called Bee Engaged, like with several E, celebrating the biodiversity of bees and beekeeping systems. And also we in PRIMA, we are devoted and attached to celebrate the Bee Day every year, as you may have participated with us last year in this celebration. This year we have created a story published on the PRIMA website and on the social media platforms about the preserving of biodiversity and how it is a great asset in the Mediterranean. And we have in this story especially highlighted how PRIMA projects, the four of you, have worked on preserving and helping millions of beekeepers in the Mediterranean region. So just to make it very short, on behalf of PRIMA, we thank you so much for this event, and we wish for more and more synergies and more collaboration and research when it comes to the bees. Many thanks again. I'm looking forward to listen to you.
Thank you, Sarah. Thank you so much. Thank you for being here and for greeting us. Is something that Ida wants to say? You are muted, Ida.
Now, can you hear me?
Yeah,
I think
yes.
now. Okay, thank you. Thank you for unmuting me. I'm so happy to be here. Thank you so much for the invitation. As you and Sarah also mentioned, I am one of the project officers at the PRIMA Foundation. And I think, very lucky, a very lucky project officer, because I'm the one in charge of 3 out of 4 of these projects. So I get to see the interesting work that is being conducted. So I get to follow what is being done with these very important and successful projects. And I think, as someone who isn't an expert, who isn't from the field, it's very interesting for me personally to be here to watch these presentations that you will make today, to have a better understanding of the role, the vital role that bees play in the environment for maintaining our biodiversity, to contributing to our food security. And as Sarah also mentioned, as PRIMA, we are always happy to see the consortium of different projects coming together. This is a pleasure for us to see. So it's very wonderful that you have organized this event together, that you have come together, you have gotten in touch with each other to create synergies, which can grow into more collaboration, to growing together. So this is something very special to see. So I'm very happy to be here. Thank you so much for your invitation. I hope to learn more things from you during these presentations, and I look forward to it. Thank you so much.
Thank you for being here with us. And I don't think there is somebody else from PRIMA, right? Okay, then maybe I could ask our good collaborator, Rosita Baumann from FAO, to give us a few words. Thank you, Rosita. Danny, did you unmute Rosita, please?
Okay, thank you, Danny. Thank you, Fanny. Well, I just would like to say thank you for the invitation to this, I think, very important event. And as Sarah mentioned already, the theme of FAO's celebration of the World Bee Day was celebrating the diversity of bees and beekeeping systems. And we all know that there are more than 20,000 bee species around this world. And also the diversity of beekeeping systems is quite amazing. And in the work of FAO, sustainable beekeeping systems is in the focus, while another group works with other pollinators. The bees is just a little bit part of all the pollinators we find around the world. And we are super grateful to this group of people to implement such important projects to support sustainable beekeeping and support our pollinators. With this, I would like to wish you not only a successful event today, I would also like to wish you very successful project implementation. Thank you.
Thank you, Rosita. In fact, for me, it's a double pleasure to have you here. First, because we are collaborating in this project with you, and FAO has a representative and we can always discuss. But second, because we are also collaborating through Apimondia. So I'm really very happy for this collaboration and honored. Okay, then maybe then, Peter, could we have your word from Apimondia, Peter? Where is Peter? Peter is over there. Danny, could you unmute Peter, please?
Hello.
Yes. Hi.
Thank you, Fanny, for the invitation. I would like to congratulate you that you connected your event also with celebrating of World Bee Day, because probably you know Slovenia was the country from where this idea came from. And therefore, each year we have more and more work to talk with different groups of people from the whole world to explain them why we started this procedure and why do we need this World Bee Day. And I'm very glad that each year more and more people would like to celebrate World Bee Day, and I think that really this is very important. So therefore, I'm very glad that also this event it is some way in connection with celebrating of World Bee Day. Otherwise, I want to be very, very long because we are almost too late. If a little bit late, if I look for this this schedule. So I wish you a good event, and I'm looking forward to hear where we are with the project. Thank you.
Thank you, Peter. Really, really good to have you also with us, and always enjoy the conversation with you. So, Mr. Ratia, it's an honor, please.
Hello, everybody. Thank you, Fanny. Thank you, everybody, for your presence. So I have good news and bad news. The bad news, I didn't prepare any speech because you didn't tell me before. And the good news will be very short. So Rosita said we have more than 20,000 different bees. And us as a representative of Happy Mondial, so representative of 7 million of beekeepers around the world, we employ only a little bit more than 20 species of bees. And they are very important for the pollination, for our human food. And we are very happy to collaborate to this project and also to this wonderful World Bee Day. Thank you.
Thank you, Gilles. Thank you so much. Thank you for being with us. Okay, then finishing with the formalities, but we have something for you now. Danny, back to you for the video.
Bee health and its impact on human health. How are we affected by the world around us? Our environment's harmony, health, and beauty are based to a large extent on the variety of its flora and fauna. Since we are part of the environment, our well-being is also linked to this biodiversity. When we speak of healthy bee communities, we refer to those having sustained species diversity over time. But how is this linked to our well-being? Studies over the last 10 years have shown 4 main positive aspects of healthy bee communities over humans. First, nutrition. Bees are the main pollinators of most food crops. Therefore, they benefit the quantity and nutritional quality of vegetables, fruits, and seeds, essential foods for our diets. Second, unique and valuable products. Bees produce honey, pollen, propolis, royal jelly, and wax, and also pollinate approximately 28,000 medicinal and aromatic plants. There is strong evidence that these products and foods improve our well-being when used as medicines and cosmetic supplies. Third, mental health. The green spaces and biocultural landscapes that bees help sustain are environments that we enjoy, allow us to connect with life in nature, and improve our mental health. Beekeeping and agrotourism in its all variants are fundamental examples of activities that contribute to human well-being. 4. Less polluted environments. Bees depend heavily on plants and are highly affected by pollutants such as agrochemicals, heavy metals, and biocides. Initiatives seeking to minimize their use to ensure pollinators' health, reduce toxic waste, and promote healthier environments for all. To summarize, diverse communities are a good indicator of healthy environmental conditions for human beings.
Great, thank you, Danny. So that was the last video that we produced in Apimondia. It's collaboration of 3 commissions, the Pollination and Bee Flora, the Bee Health Commission and Apitherapy. And it's under the concept of one health. So let's start then. And hello and welcome to the ones that they join us now. And we will start, I thought that maybe we can start from the older project. So we start with plant B. Konstantinos, it's your turn.
Okay. Do you listen to me? Fanny, I don't listen you.
Sorry, I muted myself. Yes, we listened to you. Yes, that's
sure.
fine.
Okay.
You can share your screen if you wish.
Yes. Before sharing screen, I would like to thank Fanny for the initiative. Okay, it is a very good initiative to, to celebrate the World Bee Day in the frame of PRIMA project. So thank you, Fanny, for this kind invitation and initiative that stems from also from the PRIMA initiative to try to, to collect information and explore synergies between projects dealing with bees. And of course, I would like to thank PRIMA Foundation and all colleagues and people watching this important event. So I will share my screen right now.
It's not coming yet.
Yeah, just wait for.
No problem. Yeah, it's coming.
It's coming now?
Yes, it's great. Thank you.
Is it okay? Okay. So the Plant B is a PRIMA project called the Sustainable Mixed Cropping Beekeeping System in the Mediterranean Basin. Of course, I present today's slides on behalf of all consortium, which is a coalition of 10 partners from 6 countries. So we cover a big area of the Mediterranean basin, and countries are Greece, Italy, Spain, Algeria, Egypt, and France. And it belongs to section 1 of the farming system. That was the call of 2018, under the call of the improving the sustainability of the Mediterranean agroecosystems. Due to the COVID that we were all more or less affected, PRIMA Foundation granted us an extension in order for some activities that were impossible to complete due to field visits, etc., in order to, to try to fulfill all the envisaged aims of this project. And the main objectives of Plant B is to provide a new integrated pest management tools against citrus crop and honeybees. Another important topic that we deal with in Plant B is how we will valorize well-adapted endemic honeybee species. So there is also a lot of work on the valorization of these endemic species. This is also important because we aim also to assess the potential to resist to major honeybee pests. Of course, we need and we scheduled and we organized how we will implement this system on the field. So we propose the farming system that is it takes place right now in the Mediterranean region. Since we combine citrus, aromatic medicinal plants, and honeybees within the same fields, The honey is also a key product of the project, so we try to assess its quality and safety. The socioeconomic part, and it's also very important, it's another pillar of the project. So we try to ascertain this part along with the environmental sustainability. And of course, try to see and explore actions to promote these new tools or new products or new knowledge found within Plant B. Plant B comprises of 7 work packages. So there is an interrelation, of course, between them. The first work package has to do with the management of the project and the establishment of tools that are essential. Fabrice was here in the kickoff. We are all in line with PRIMA Foundation and with the rest of colleagues. So there is the project management committee, several tools that are important, such as the stakeholder advisory board, and of course the project workplace that is important through our website, some SharePoints in institution, and of course the reporting platform that PRIMA has established, with which we try to upload the work and the deliverables, etc. And to go to the pragmatic work, as I said, in the second work package, we deal with the development of these tools that are biological control and IPM tools to control pests in citrus, aromatic medicinal plants, and the honeybees in their colonies. So there is a lot of experiments going on within Plant B. For example, on the left side, we see a picture with trials with entomopathogenic nematodes for the control of the med fruit fly in a case study in Arbolida region in Greece. And on the right side, we see how a comb is divided with the bees in order for some experiments to take place. Another part that is important is how we try to deal and to provide new or let's say enhanced solution in the management of Varroa. We all know the importance of Varroa. Even I'm a chemist, I have managed to understand what is Varroa for bees. So there is a lot of work from colleagues in order to use semiochemicals to confront with Varroa in honeybees, and there is a lot of work behind this. So colleagues test several non-volatile compounds, and in order to assess how these will be effective against Varroa. And a review article was also published from our colleagues, and there is also work being done right now in order to, and results are produced, and more of them will come into reality this year in order to see which were the outcomes of this semiochemicals testing. Galleria melonella is also needed to tackle it, so integrated pest management is also very important. And colleagues within Plant B study the effects of these new resistant wax combs. They try to develop new combs that will be resistant to Galleria melonella. So it is an activity that we really try to build upon and try to produce resistant beeswax. And the first results are promising. And in this direction, there is a specific protocol with the treatments, and of course, essential or fixed oils are used in this direction. Last but not least, within these IPM tools and the work package too, is that in Plant B, we also tried to see how precision livestock farming strategies will help beekeeping. So colleagues in Italy are testing and already assess bee systems, such as the 3B system, which is used in precision beekeeping. And of course, respective protocols are developed and work is progressive in order to have tangible results on this point. Though the first part regarded the bees, but of course, IPM tools aim to be developed against citrus and aromatic medicinal plant pests. So in Spain, cover crop and companion plants are have been installed, and they are used as means to enhance biological control. This activity takes place in mandarin orchards in Spain. In Egypt, the efficacy of the entomopathogenic nematodes is assessed against the fruit fly and other aromatic and medicinal plants in several laboratory bioassays. And in Greece, as I showed before, there are tests and experimental protocols applied for the efficacy of the entomopathogenic nematodes of Bacteriophora and others against the Mediterranean fruit fly. So there is a lot of work that has to do either with the IPM tools of against honeybee pests, and of course respective IPM and biological control tools against citrus and aromatic medicinal plant pests. Within Plant B also, we tried as a continuation of a previous work that was implemented in olive groves to further enhance and optimize this impact assessment procedure, which, as I said before, it was developed to assess the environmental impact in previous projects, but now it was implemented and adjusted and further enhanced for the scope of Plant B in citrus orchards. So a lot of work was conducted on the parameters of this impact assessment procedure, which is actually it is a software tool which can be used in order to assess this impact, the impact of all these activities within Plant B. It has been already developed. It is functional, and it has been reviewed, and it was implemented in the orange orchards in Argolida in Greece, and further implementation of the app is taking place in other orchards in the rest of countries. Final aim of the app is to serve as a tool for the targeted advisory services to citrus growers in real time, because real-time data can be incorporated in this tool, and it can really depict the sustainability in terms of metrics. And this is how the environment of EAP is a very functional tool. Several parameters, as I said, can be introduced, and in the end, very significant results can be derived. It is also important to mention that, as I said, a lot of colleagues work within Plant B in order to conserve and valorize the endemic honeybee subspecies. So this takes place in 3 countries, in Algeria, in Egypt, and Italy, with specific species, such as in Algeria is the Sahariensis and Intermissa Apis mellifera, in Egypt is Apis mellifera lamarski, and in Italy is the Apis mellifera ligustica and Apis mellifera sicula. So there is a lot of work, and in the beginning it was important to establish a sperm bank for the Saharan and for the Italian bee, so sperm was cryopreserved. The next step is to apply that on the field in order to valorize the native honeybee subspecies. So there are promising results with some of these honeybee subspecies, and how can we valorize them, because some of them can be resistant to some pests, so it is important to valorize them and use them. I will not say a lot of details about the experiments. There are several protocols, such as the hemolymph extraction and then the quantification of the protein. And of course, this work without networks will not have the same impact. So there is local networks that are already established in order with local beekeepers to enhance these subspecies and to share the knowledge with the people that they will use them, the beekeepers. The 4th work package are the actual case studies in which the citrus orchards are combined with the aromatic medicinal plants and the honeybee. So you can see on the left side some pictures from Greece with the positioning of the beehives, the stripes of land in which aromatic medicinal plants are installed, and the citrus orchard. So case studies take place in 4 countries: in Egypt, in the Luxor area with oranges, in Greece, in Algorida region that I've mentioned, in Spain, in Valencia, and in Italy, in Sicily and Campania. The case studies, of course, integrate the citrus, the aromatic medicinal plants, and the beekeeping. All baseline measurements, and after the establishment, because the first year was the pilot, let's say, year in which the first aromatic medicinal plants were installed. So all these measurements have been conducted, and honey was collected the first 2 years, 2020, 2021, and now we are collecting the honey of the third year of the project. And of course, all the plant protection activities that take place are also recorded in order to feed also the impact assessment procedure. I will not stay so much in these slides. Some pictures with what is the concept, that we have some control fields and some other fields in which we apply the plan B scenarios, and then several results are derived that have to do with metrics on the field, but also with the honey as an end product. The first results that were also introduced and were also presented in the IOBC of 2022 Congress in Efplio in Greece, showed that a significant higher number of pollinators and natural enemies were identified in citrus field margins. And of course, wild bees were also registered and evidenced, and some of them are also shown in this slide, but no correlation between the weed vegetation or the flower cover and the insect abundance or diversity could be proved. In Egypt, several case studies also take place. More specifically, again, we have citrus orchards, which are the control, in which none tool is implemented, or even only honeybees are positioned there. And of course, we have the fields in which several interventions took place. This is more or less the rationale within Plan B. We have the control fields and other fields in which we apply the scenarios in order to assess potential differentiations. The only individual point that we need to stress is that in Spain, since there is no beehives were placed, because usually it's not an everyday practice to position beehives within citrus orchards. So no beehives are placed in the case studies in Spain. And of course, within these field studies and case studies, the management and the impact of the ground cover on management on the natural enemies is recorded. For this reason, several techniques are used, such as the vacuum machine for suction sampling or pitfall traps. In the fifth work package is the work package that has to do more or less with the chemical analysis. So honey and the citrus crops are analyzed. Citrus crops are analyzed for pesticide residues, but honey is also analyzed for pesticide residues, but also for several of the points that are also mentioned in the honey guideline with regard to its quality, such as the hydroxymethylfurfural or diastase enzyme, or other metrics such as electric conductivity, but also for the bioactive compounds. The latter is done because we try within Plan B, even if we have, let's say, homologous type of honey produced, because the majority of them are citrus dominated. Nevertheless, we try through the chemical analysis to unveil chemical markers that might differentiate honeys between countries. This is conducted within a battery, as we say, of analytical method that has to do with the bioactive compounds, but also with the analysis of trace and macro elements, because this is also an important pillar for the differentiation of the botanical origin of samples. The pesticide residue analysis did not show for the first 2 years something, let's say, alarming, because we did not observe any exceedance of the MRLs on the honeys produced, and we cover a broad range of analytes in our analytical methods. Similar was the picture for the citrus commodities. And of course, we analyzed and pollen and walks, since it's theoretically and not theoretically, practically and realistically are matrices that are more, that bear more contaminants. And what is important that I mentioned before is that even though we deal with similar types of honey, citrus dominated, we did evidence differences that are related, of course, with the geographical origin, but also with some differentiations among the field orchards, because aromatic medicinal plants were there and some different kinds were installed in countries, or different pre-existing aromatic medicinal plants existed in the nearby areas within the foraging area of the bees in the case studies. A questionnaire was developed. It is very important in order to assess the environmental and the socioeconomic sustainability of the project within this, let's say, complex farming system, is to have a questionnaire with the registration of all parameters and all information that will be important. So there was a lot of work by colleagues behind this questionnaire and this data entry file that we call, in order for all people involved, citrus growers and beekeepers, in order to fill this information that is then important for the colleagues who perform the socioeconomic analysis. Again, we have some good results, and I think that within 2022, more results will be published with original research, or even the assessment of the impact assessment procedure, and of course, the socioeconomic and sustainability issues. But without dissemination and without work on this aspect, a lot of projects will suffer without diffusion of the knowledge produced. So we have a very, let's say, functional website. You can visit it. You can be, we are also active as a consortium on the social media. So there is a lot of work uploaded, and the Plant B website is updated in order to be in line with what is produced within the project. And several, an annual conference was conducted in order to show the progress and what we were planning to do about the analysis on within Plant B. A lot of work is produced on leaflets, on descriptions. There are interviews among people who work within the Plant B. And of course, a lot of information, I need to stress it again, is on the website of the project. And but there is only a website. There is also a lot of work of people who try to disseminate the knowledge. They perform field visits, people that are responsible from Egypt in the communication of the project help us a lot in order to collect all this material from fields and make interviews, announce Plant B activities within the site in agribusiness forums, or even in specialized magazines among the countries. I would like also to show you some videos. So this is how we valorize the Egyptian bees grain.
احنا حاليا بنعمل الملكات الزياده على اساس ايه؟ كده دخلنا 10 ملكات. دخلنا 10 ملكات الفروض اللي احنا عملناهم، وهنا بنخزن الملكات دي وحاجه فشلت نغيرها نستبدلها. حاجه ماتت من الملكات العذاره اللي احنا مدخلينها.
So this is a video where colleagues try to valorize the Egyptian bees queen in the specific reservation place in Egypt. The second video is from the orchards in Greece, and we see an apis that is on the citrus. So there is a lot of material that demonstrates the work being conducted. And another important video is from our Italian colleagues on how the queen is born in Italy. So that was from my side. Again, thank you, Fanny, for the initiative and all the people from the PRIMA Foundation that support the project. And of course, Plan B Consortium, our institutes and bodies. And also, I hope to integrate with the rest of the projects and be happy to answer any questions after in the QA session. Okay, thank you very much.
Thank you, Konstantinos. It's really very good to hear a few things about the project again, because this is the second time I hear, but every time it's even better. I'm very happy that we have some things, there are similar ideas, different approaches, and they could be even supplementary. Yeah, I'm really very interesting to see your results and maybe talk about more in another occasion. I really like the cryopreservation stuff as well.
Yeah.
Cool, very nice. So maybe we can quickly continue with the second project in the order, which is MediBees. It was started before us. And Antonio or Alice, I don't know who's going to start. You choose between you, please.
Yeah, okay. But
no, it's Antonio.
I suppose that I start, and after comes Alice giving some more details about some specific work packages.
Thank you, then.
Yeah, okay. So thank you very much to the institutional representatives that are present here. Thank you very much also to you, Fanny, for this celebration. And the reality, this is the second time we celebrate the WBD Day this year, because the first time was in Pantelleria when we signed an important document about the protection of wild honeybee colonies. Well, going to the project, our project, as you can see from the title, is about resilience to the climate change of the Mediterranean subspecies, with a focus on the genetic background of those traits, and also specific tasks on the products of those subspecies. Well, the coal we wanted to respond was the farming system 2020. So it was just before the COVID pandemic. And well, I copied a few sentences from the coal. As you can see here, actually coal was not really focused on the honeybees. You can see livestock, breeds, meat, milk. So it was something more general. So we doubted about the possibility to participate, but well, after some indecision of the first moments, well, we decided that we had very good arguments, very good elements complying with the coal. Well, one of them is that the animal we are speaking about are the bees, that are considered the third most economically important animal in Europe, not only for their direct products, but also for all those the added value of the externalities the bees and the beekeeping gives to other productive sectors, mainly the agriculture and also to the environment.
Excuse me, Antonio, we don't see the presentation. Is there a presentation?
Yeah, there is a presentation. Yeah, sure. I
but
forgot.
we don't really see it.
Yeah, sure. I forgot to share it. So sorry.
But that's all. That's fine. That's okay. I thought there is something wrong with the Zoom.
Yeah. Sorry, I'm trying to.
Yeah.
To
with the green button.
Yeah, sure. I know which is which, but yeah, I think now
it's coming now.
Yeah, you should see it properly, right, sir?
Yes, we do.
So okay, so I go back to the first slide. You see, there were some keywords that didn't have actually much to do with bees. So we decided to participate anyway, because we have the bees that are considered the third most economically important species in Europe, and which they are also providing a high added value to the agricultural production and to the environment. Well, and actually we don't have breeds, but we have subspecies, and the Mediterranean concentrates approximately the 40% of the subspecies diversity of Apis mellifera. So there was a sort of an heritage to protect, to promote, and also to improve. So we decided to participate. The consortium is made by 8 participant countries: Portugal, Spain, Italy, Malta, Turkey, Lebanon, Jordan, and Algeria. 8 are the participating countries, but 9 are the partner institutions, because we wanted to have among the partners the Jordanian Beekeeping Union. So Jordan is represented twice as a research institute and also as a stakeholder. And 10, so the numbers of our project are 8, 9, and 10. 10 are the Apis mellifera subspecies that fall under the territory. These are, let's say, not controlled, but anyway involved, that is involved by our project. So Apis mellifera iberiensis, ligustica, siciliana, rutneria, anatoliaca, caucasica, meida, syriaca, sahariensis, e intermissa. Well, in general, what we want to do is to understand, to decipher the genetic background of the Mediterranean subspecies. We want to study the adaptation of those subspecies to the specific environmental conditions, and to detect and understand the basis of their differential resilience to climate change. We have more specific objectives. We want to make a genetic mapping of all the subspecies that we are going to study, and with a specific attention to the aspects of their adaptation to the Mediterranean environment, and even more to the climatic changes. So we are going to make experiments on their resilience or tolerance to heat stress, to drought, to UV exposure, and also we are going to consider the resistance, the differential resistance to parasites and pathogens. Well, we included also actions directed to the valorization of Mediterranean products, namely the honey, and also the byproducts. There are normally the byproducts produced by the apiculture industry are treated as a waste, but we want to reuse them in some way to give an added value to the and an added income to the beekeepers. This is the structure of the work packages of our MedBees project, which started in the beginning of May of the past year. So we are have just started the second year. There are 6 experimental work packages that are emphasized in bold, and other 3 work packages. The work package 1 is basically intended to collect information and to plan the actions. Work package 8 is about all the communication, dissemination activities, website, social media, and so on. And also we have a work package which is intended to coordinate the consortium. Well, the experimental work packages are the ones in bold. Work package 2 is for the collection of sampling in all the involved countries, some bee samples that are intended to feed the activities of work package 4 and 5, which are basically molecular activity, genetic activities, and those are actions that Elise will explain you in more detail. We have also in the same work package 2 the preparation of apiaries and the assessment of how the colonies work in the different conditions. So we will consider the colonies of the specific Apis mellifera, of the Apis mellifera subspecies that are specific of each country in comparison to a control. So field trials. We also have laboratory assays that are going to be made in caged bees reared in incubators that are exposed to stressors. And going to the work package 6, we have activities about honey valorization. We want to promote, or better to help the beekeepers promote their productions, or even better, the honey produced with the local subspecies. And work package 7 is about the use of beekeeping byproducts. Well, this is how the work packages interact. Well, we have basically 3 lines. One line is about the honey. We are going to have sample collection throughout the involved countries, and those samples will be submitted to traditional analysis, sensory analysis. Panels of sensory analysis will be created in each country to help the beekeepers promote and valorize their honey. And there is also an action intended to a molecular study of the honeys themselves in order to detect the botanical origin and also to study the possibility to detect or identify frauds. Also, we have here byproducts. Mainly we think to use the byproducts coming from the beekeeping activity to produce media that can be used in the agriculture, maybe in the production of high value agricultural products like flowers, for instance, or maybe in horticulture. And well, the bees, these are the work packages dealing with bees. And well, briefly, the general idea is that we are going to study the bees at social level, at individual level, and at the genetic level in order to identify in each subspecies genetic markers of a differential resistance to the climatic stressors. And when we have those markers identified, maybe we also have or can give the beekeepers a tool to breed their own stocks, their own local stocks for an improved resistance to the climatic stressors. Obviously, our project is not as advanced as the project we saw before. We have just started, so we basically the experimental activity. Most of the experimental activities are just at the beginning. The activity that's more advanced is the work package 1. We submitted a questionnaire to more than 1,000 beekeepers in all the countries, asking them questions that were intended to characterize the beekeeping activity in each participating country, and also to identify the main problems the beekeepers experience, especially the ones related to the climatic change. And well, we have the plan of sampling that are intended to provide bees for the genetic work packages has already started throughout the involved countries. We are very close to establish the experimental apiaries for the field tests, and some of the preliminary assays, laboratory assays, have already started, and they are preliminary to the preparation of a protocol that will be applied at each laboratory level to make the studies with the bees from the local subspecies. The activity on sensory analysis has already started, and there are also preliminary trials on the use of byproducts, and also communication activity is already there. I go through quickly on few slides to show what's going on in those work packages. Here is an example of the questionnaire. This is just the first page of the questionnaire, and few of the results that are limited to 3 countries only, but they were submitted. The questionnaire were submitted to all to the beekeepers in all the countries. So we have a range of a great amount of data to analyze and publish shortly. Well, we have a sampling plan. We designed transects in different countries that have mainly, I would say, they are transects that have a geographical meaning. You know, for Italy, for instance, the transects are from coast to coast. In the Iberian Peninsula, they were drawn north to south. Similarly, in Turkey, they were drawn north to south to catch both the plains and the coastal areas, and also the high mountains that are in the middle. Well, it is interesting that some colleagues from other countries, and namely Iran and United Arab Emirates, showed interest in our activity. So they think they agreed to participate to many of our activities with own funds, you know. So our group is enlarged with respect to the official partners belonging to the project. Well, this is an example of the apiary that we are going to prepare in each country. We are going to have a group where bees from the local subspecies are reared, and a control group which is equal throughout the whole experiment, which is a group of Ligusica bees. We in those apiaries, we are going to measure for 2 years the development of the colonies, the amount of honey produced. We are going to measure behavioral variables like the swarming activity, the gentleness, the hygienic behavior, things like that, the foraging preference, and at the end we will make an evaluation also of the reproductive parameters of the individual queens and drones. The laboratory trials are intended to study the main stressors that we expect to arrive, or that are already arriving, we are already experiencing with the climate changes. And this activity involves transcriptomics also, and studies on gene expression. This is an example of the sensory analysis panel we met in February last in Jordan. It was the first meeting in presence, and we had a very short course on sensory analysis of honey. And another gathering, which will be much bigger, will be in Bologna the coming July. This is just an essay on what is going on with the hive byproducts. They are processed, homogenized, and analyzed at first as a first step to understand which is the nutrient content that might be useful in a possible agricultural usage. Communication activity, we have our website. It is in construction, you know. It's we are just at the beginning. It's hopefully going to be updated in a very short time with many information. We are on the main social media, as you can see here. Well, so as you can see, we are rather enthusiastic about our project, and also, well, we are aware that it is an ambitious project also. So we have at the left side, you know, what the research proposal often look like, which are amazing, nice, articulate, but for sure we don't want to go to the right side and make such a sort of contraption. So we wanted to keep to the plans and to keep with the same enthusiasm and this activity as just as described in the project. So thank you very much for your attention. My regards to everyone, and not only from me, but also from all the consortium that is composed by approximately 35 colleagues. So thank you very much.
Thank you, Antonio. Very interesting indeed. And we also share partners and people.
Yeah, sure.
Like Lisa from Jordan, and he's in our advisory board, and Nuruddin, that
yes.
is your partner, which is also our partner. So we share a lot, also common interest. That's really great. Although we run very late, I ask everybody to be patient, because everything is really so interesting, and it's good for us to understand what we are doing and what we want to achieve. So shall we move? Could you please stop sharing, and then we could go to Alice Pinto.
Yeah. Sorry, let's try to.
Dear Alice, get ready.
Hello.
Okay.
Okay.
Yep. There you are.
Need to find. Okay. Good afternoon, everybody.
Would you like to make it full
Full screen?
It is.
Oh,
it is.
Okay.
That's fine. Yes. Great.
Good afternoon, everybody. And I would like to start by acknowledging Fanny for organizing this meeting. I think this is a good platform for exchanging ideas and collaborations.
My pleasure.
We hope we can collaborate after this meeting. And I would like to thank the representatives of PRIMA and Apimondia and Apimondia, right? Oh, and FAO, yeah, for taking your time to be here to listen to us. I'm going to talk about work package 4, which is about the genetic studies. And I'm going to be very brief. Actually, some parts Antonio already explained. And why we decided to talk about work package 4, it's because work package 4 is a central work package in MediBees. If we accomplish this work package successfully, we will answer to the main goals of MediBees. And it's also central because at the end of the project, we hope to have a good understanding of the diversity patterns, genetic patterns across a wide geographical area, because we have many countries, and especially we are representing 10 subspecies. And we hope to have a good understanding of the genetics, because genetics and diversity is the raw matter for selection and therefore for adaptation to a changing world that is changing very rapidly. And I think we all agree that diversity is the base of adaptation, and without diversity, there is no, the bees are doomed, right? And there will be no projects. So we share some common interests. I saw the presentation of Konstantinos, who showed that they also want to valorize endemic subspecies. And this is also one of our goals. So these are the objectives of Work Package 4. We will develop, resequence 9 subspecies because Iberiensis, we have already many genomes. So we will resequence the 9 subspecies that are represented in our study area. And from the whole genomes, we will develop SNP sheets, so sheets with markers for the several subspecies. And using the data that we will generate from the whole genomes and then from the SNP scans that we are going to run, we want to identify candidate genes for local adaptation, especially adaptation to climate. We also want to, with this data, to assess the conservation status of Mediterranean honeybee subspecies. We all know that there are many subspecies that have been threatened by introgression from commercial breeds. We want to have an idea how spread is this introgression, and to see how genetic integrity of the native subspecies that we want to preserve and to valorize. And finally, we want to identify genetic variation that is linked to stressors and diseases in an attempt to understand the genetic basis of adaptation to these stressors and diseases. And this will be objective is connected with the Work Package 2, where we are going to have the experimental apiaries that Antonio talked about. So we are, at the moment, we are sampling, like Antonio explained, and we are sampling across a very wide geographical area. We are building a comprehensive and spatially referenced collection that is collected across environmental gradients, and we have very extreme conditions, like climatic conditions, like Sahariensis that lives in the oasis in the desert of Sahara Desert. And we have, for example, Caucasia, Apis mellifera Caucasia in Turkey that occupies mountain areas, so cold environments. When we are building this collection, we have in mind that we want to capture, on one hand, the variation covered by these subspecies, the genetic variation, and also we want to cover environmental variation, because to find adaptation to environment, we have to have a good coverage of both. So Antonio showed this slide, and this is, I selected this slide because Turkey is the country that has the highest number of subspecies within our consortium. And when we designed the the sampling transects, we had in mind that we want to cover these environmental gradients latitudinal by designing transects across latitude and longitude, and also across mountains. For example, for Caucasia and Meda, we want to cover altitudinal gradient in order to find the genetic basis of adaptation to altitude, which is also genetic adaptation to climate. This is the sampling sizes that we have and the subspecies that are included. It's a very ambitious sampling. We hope to get about 2,000 samples that we want to sequence the whole genome. So we have the subspecies that are represented in the countries of the partners, and we have also the collaborators, like Antonio mentioned. In addition to Iran and to the Emirates, we also have Egypt. And we are open to collaboration with you, because it will be interesting to add more Mediterranean subspecies that we are missing several. For example, in Greece, there are like 3. So the more, I think, the better we will be able to understand what are the genetic strategies or solutions that bees find to adapt to the different environments. So in the work package 4, we have several tasks. We will start with the whole genome sequencing of all the subspecies, and we will use a battery of tools to scan the genomes to find these the genes that are putatively involved in adaptation to climate and other stressors. Then we hope to find these variants. We will understand where are these variants located, if they are in coding regions or controlling regions of the genes. We will use then this variation, the SNPs, to include, to design and to build a ship for the Mediterranean subspecies. In this ship, in addition to include candidate genes, SNPs that are marking candidate genes for adaptation, we want to include SNPs that are specific of the different subspecies, so we can use this ship to identify the status, to identify the status of each subspecies, to look at how introgressed they are across the Mediterranean. We want also to include in this ship SNPs that are linked to immunity genes and other SNPs linked to stressors that have been identified in other studies. With this ship, then we will screen a higher number of samples to look for the diversity patterns, the neutral. When I say neutral, these are non-selective for the variation that is not under selection. And we want to look at the patterns that are under selection across the geographical area, to look at the conservation status of each one of the different subspecies. And also we want to understand the phenotypes that we are gonna measure in work package 2 in the experimental apiaries. And I think that is it. Thank you very much for your attention, and I hope we can find some interest, some interest in common
place.
Yeah. Thank
common
you very
place
much.
to meet.
Yes. Thank you very much for
Thank
your
you, Alice.
attention.
Thank you so much. Thank you for being here. Thank you for sharing this work with us. It's really great. So before
Fanny,
yes.
Just one word. I wanted to let everybody know that Alice and myself are here as a representative of our coordinator. So our coordinator is Raquel Martin Hernandez, that's a friend of many of us. So I wanted to mention that.
Yeah. Yes, sure. Thank you. I'm really very happy with Raquel and Alice because they also agreed to collaborate on our questionnaire. So it's another way that we are doing synergies. Just we didn't show them yet.
Yeah.
Okay. So before we continue, just to break a little bit the monotony, we suggest that we should have a group photo first, if you agree. So please all have a gallery here, or have your cameras on if you wish. If you don't wish, that's fine. So I can make a few screenshots, and I cannot take everybody in one. I'm still waiting. Yeah, okay. Yes, you can smile, you can say bees. Yes, I think we have everybody in one picture. I don't have any other, an arrow pointing. So we have everybody in one. This is really good. Okay, thank you.
Fanny, can you then send that picture? Because if everybody agrees, we would like to post it in our website.
Yes, I will. We are already on air anyway, so everybody can see us and listen and hear to us. So we can show them our nice faces as well for a longer time. Okay, so we have a very short video before, just for a short break. And Danny, could you share, please, the one with the farmers, please? Because I made a mistake and I showed you, or we showed you, the video by Yapimodi in the beginning while I had it as a surprise for the end. So now we go a little bit backwards. So this is by FAO. No. No, that's the beekeepers. No. We need the FAO about the farmers. No. The devil of the technology is doing something on your computer. And until Dan is finding the video by FAO, I was saying that when I was invited by Raquel Hernandez to participate in the Medibees project, I was too busy with our own project to prepare. So I said, no, thank you very much, but I can. And now that I could see the countries and the partners and the Balkan Peninsula and Greece was out, I was really feeling very bad. I regret that one. And you are a lovely consortium. Thank you, Danny. Let's see it. Very short, very nice, very much to the point. Good. Then we continue with the Safe Agrobi project. That's our project. And Clemence will start sharing the first part. We broke it down in 3 parts. We are not going to tell you everything about everything, but we will concentrate. So yeah, can we have it in? Yeah, great. So I will just talk over the first 3 slides. Yes, can you make this one like a slideshow? Thank you. So these are the countries and the partners. We have 2 partners in Greece, 2 in Slovenia, 2 in Croatia, 2 in Italy, 1 in France, 1 in Algeria, 1 in Cyprus, and 1 in Lebanon. And we are really very happy with this consortium. We focus on the resilience of the beekeeping based on the pollination services by bees, but also on the use and adaptation of the local subspecies. Next one, please. Yes, Clemence, can we have the next one? Yeah, okay. Here you see a general concept, very nice drawing made by Giorgio. Thank you, Giorgio. Where it shows that we will collect all this information from the agro ecosystem where everybody's part, like the beekeeper, the landscape structure, the honeybees, the pollination services, the weather, of course, and put them all under in a platform where we collect observations, data from the observations, but also data from precision apiculture systems for intelligent systems. And then the task of our modelers from Italy will be to create models to explain the interaction between all these factors and variables, but also maybe to predict what is going to happen later on in order to take to do mitigation measures and to support, of course, beekeeping. Next one, please. So that's how we visualize the whole project in 5 research working groups. One is the pollination, the climate change. The second one, adaptation and the performance of the local species. The minimizing bee losses is work package 3. Precision apiculture tools in work package 4, that they are really very close, interconnected. And then, as I said, everything will go to the platform. And then the modeling tools will give us maybe a good idea of what is happening. And of course, the coordination, the management, and the training. The training, we saw it and dissemination as something around, like grasping the whole project, because it's a lot of training going on between the partners, but also with the citizen scientists that they are helping us to collect data, beekeepers or farmers. And we would like to work a little bit more on the citizen science approach. Also, what we are very glad for is that we have an advisory board where you can see the names Nicola Kezis, Jill Saratia, Anna Gaida, Danny Obeid, and Nizar Haddad, and the key stakeholder representatives from Epimondia and FAO. From Epimondia is Peter Cosmos, and from FAO, Rosita and Amdam. And we are really very, very happy to have them with us because they really guide us and we discuss with them every so often. So yeah, that's the general idea of the project. I don't want to stay more. I would like to call then Clemence to continue, and she will explain to you more about the work package 1. Clemence, back to you.
Yeah, thank you, Fanny. Hello, everyone. I'm very happy to be with you today. I'm Clémence and I'm from France, and I'm as a postdoctoral fellowship at the EGCE lab in Fabrice Requier team, and I'm here to help with the coordination of the work package 1 from Safe Agrobi project. Work package 1 is about understanding and conserving the provision of insect-driven pollination services. And in work package 1, we have 3 tasks. We have a task about pollination experiments. So for this, we are performing some pollination experiments in 5 countries. And we are doing this in major crops, such as apple orchards, or this summer, we will also work on sunflowers field. We have a second task, and with this one, we are working about with honeybee hives, and these hives are equipped with scales in order to determine the research space optimal carrying capacity of honeybee colony for specific crops. And we have a third task, which is about climate change impact on crop pollination services. And so you can see that all this work package 1 is focused on the pollination services provided by insect. But so for this subject, we miss the opinion, the expectation, and so on about pollination services from both farmers and beekeepers. So to have this, we designed an international survey. So we built 2 questionnaires. They both have about 25 questions. And so we built 1 questionnaire for farmers and 1 questionnaire for beekeepers. And the aim is to collect opinion and expectation about pollination services. So in both questionnaires, you will find some question about the socioeconomic situation of the participant. You will find some question to measure the knowledge about pollination needs of various crops. We will also know more about the habits of the collaboration. For instance, do beekeepers collaborate with farmers, and do farmers collaborate with beekeepers? We also have some question about the opinion and knowledge about climate change effect on pollination services. So the questionnaire will be disseminated in many countries. So of course, we will disseminate in countries that are involved in the work package 1. So we will have participants from Algeria, Croatia, France, from Greece, and from Slovenia. We are also hoping to have other countries to participate. For instance, we hope to have answers from Lebanon and from Cyprus. Thanks to a collaboration with countries from the MEDIBIS project, we hope to have participants from Portugal, from Spain, and from Turkey. And we also are trying to disseminate both questionnaires in Italy, in Jordan, in Morocco, in Malta, and in Tunisia. So you see, we are hoping for a very broad dissemination between many countries. So both questionnaires have already been translated in 7 languages, and we are still hoping for more translations. So for today, some countries already have started to disseminate both questionnaires, and we are starting to get some answers. So from Jordan, we already had more than 40 answers from beekeepers. With Portugal, we have about 70 answers from beekeepers and 30 from Greece. And I'm happy to say that we have more than 100 answers from French beekeepers.
Bravo.
Thank you. It's mostly thanks to beekeepers, not from, but yes. And we see that it seems more difficult to have answers from farmers, maybe because most of our networks are beekeepers, but it's a great challenge to have both point of view. So we need to be careful to have as many answers as possible from farmers too. So about what we are expecting. So we built the questionnaire during winter. We had some help from Stratos Michalis from the Hellenic Agricultural Organization. So for now, the questionnaires have been translated in 7 languages. And today we are working on the questionnaires dissemination. So the dissemination is supposed to end at the end of June. So we will be able to start to get our first results and see what's happening in each countries and in each sector. And we are aiming for a second round of dissemination by the end of the Safe AgroBee project. So thanks to this survey, we are aiming to have a comparison of both beekeepers and farmers' opinions. It will be very interesting also to have a country point of view comparison thanks to a broad international dissemination. And thanks to 2 rounds of dissemination, we are hoping to have maybe a time evolution, but the timeline is quite short, and at least an idea of the reproducibility of the answers. So that's it for my survey dissemination actuality. And of course, thank you to all the participants, people who have participated to the questionnaire construction, of course, to the many translators involved, and to each people who is involved in the dissemination of all these questionnaires. And of course, a great big thank you to all beekeepers and farmers that already have participated to the questionnaires, and to the next participant who will soon answer to our question. So many thanks for your time, and I will now let Work Package 3 have a presentation.
Thank you, Clemence. Thank you so much. Well, probably you realize, everybody realized that we swallowed the Q&A part because we are already late. And we will continue with dear Dalida, and she will talk about the work that is carrying on microbiotes and microflora of the bees in Lebanon. So Dalida, please. And maybe I should mention that we should probably think to extend the time for the questionnaires, Clemence, because we still need some translations. Maybe we can take it until the end of the summer.
Yeah, probably, because both farmers and beekeepers are in so hurry and working so much for now. So probably.
Great. Yeah. Thanks. Thanks a lot. Dalida, please. Dalida is muted.
Hello.
Hello. Good.
Well, I'm gonna introduce the work that we are conducting with the IRAL, the Lebanese National Research for Science and Agriculture, and the Lebanese University on the behalf of the Lebanese University, Dania Abed and myself. We are interested in studying this cell-to-cell interaction an impact of inhibitory metabolites released by endogenous microbiota in order to ameliorate the biological security of beehives in apiculture. So the problem that enhanced us and that induced us to conduct such work is, first of all, the presence of many diseases inside our beehives, and we need urgently some IPM or control measures, biological control measures that could help us control those diseases and pathogens without harming the biodiversity and the bees. So first of all, we made, let's say, the first part of the project was made. It consisted on studying the microbiota of the bee guts and hives inside the hive, let's say. And we saw that they contain numerous symbiotic bacteria that secrete bioactive molecules, between brackets that we can name antibiotics. So, and we observed later that they play a crucial role as antagonists against other pathogens such fungus, viruses, bacteria, and parasites. We did a huge screening, and we chose the actinobacteria, the actinomycetes, basically, because they hold the capacity to secrete secondary metabolites with antimicrobial activity against bee pathogen. So, how did we proceed? First of all, our experiment was based very simply in a first step. It was for the pre-culture. So we have our own stock of culture. We did isolate the toxicogenic fungi, such as Aspergillus species, Ascosphaera, also Nosema, and Penibacilli larvae, and etc. Many. We did many. We have many bacteria in stock. The second step was the isolation from the beehive, from the bee gut of the bacteria. Also, this technique was conducted in a very delicate way, because as you all know, that it's very dangerous when, like Aspergillus is a dangerous toxic fungi, and it requires a specialized lab to be studied. So after screening those pathogens, we obtained, we isolated from bee samples some Penibacillus larvae responsible for the American foulbrood disease, Melissococcus plutonius, the one responsible for the European foulbrood, Aspergillus species responsible for the stone brood, and Ascosphaera apis chalkbrood. So those were our 4 main pathogen, and we want to study the effect of the actinomycete metabolites, let's say, or actinomycete as cell-to-cell content, cell-to-cell interaction, or cell-to secondary metabolite interaction. The first type of preparation was conducted at IRAL also. We made many test, as you can see here in front of you. So preparation and the result, we got very promising results that are leading now our work in the field. So we want to take those actinobicetes as such without taking their metabolites, secondary metabolites, and we are aiming this month to introduce them to a multiple variety of hives, beehives, variety of species, if we can see. And we want to observe the effect of the actinomycete in helping the bee fighting those diseases. So, but before, also we need to do a gene amplification by PCR for the isolate of the bacterial disease, whatever it is, Penibacillus, Melissococcus, or for the actinomycetes by himself. Second, we need to do purifications for the aliquots, and third, the DNA sequencing, 16-strand RNA. And PCR and DNA sequencing help us to do microbial identification, a very good one. Second step, also, we are intending to do the proteomics. Let's say we mix, as you can see here in the slide, we mix the yeast extract, peptone, tryptone, dextrose, etc. With the mixture, we put the actinomycetes, and then we observe their in lab condition, their interaction. So
excuse
the protocol.
me, Dalila.
Yes.
Because I still see slide number 2. But I've seen them before going down, but then somehow then on my screen went again number 2. So could you please just go a little bit again? Maybe something happened. I don't know what.
Okay, can you ask please Danny, because he has the control.
Yes, he can hear me. He has the control. Danny,
I'm not
please.
the host.
Yes. Yeah, please continue, because we hear that. We've seen that before. I've seen it before. Can you continue?
Yes, okay. So
Danny, can you continue also?
Okay,
yes.
I don't have many things to say. I'm in slide 12, if you want, Danny. So basically, the first part of the work consisted on the lab conditions, and but to get a cell-to-cell results. And the second part, which will be conducted very, very soon, this month, I think, it will consist on introducing those actinomycetes inside the hive directly in a special way, in special protocol. We have many ideas in order to observe if really those actinomycetes or their metabolites are able to help the bee fight those 4 main pathogens or 5 main pathogens, such as AFB. So if you want to know, this is a little bit simple explanation of how we can do the proteolytic and heat treatments, and then we do ultrafiltration, fractioning, and concentration. Then we do the protein purification, and finally protein sequencing. So these protein sequencing will help us in concluding the secondary metabolite effect directly. In conclusion, this research study will provide a promising developed strategies to counteract pathogen diseases, as you can see. And such biological method evaluates the importance of the secondary metabolite that act as antagonist on pathogen. But until now, we have, let's say, around 200 secondary metabolites. So we are reducing their number. We are trying to reduce their number in the lab in order to be able to choose some 10 most important and effective. We will be based on their effectiveness, if we can say this. to include them in vivo or in the hive directly. Our study also aimed to ameliorate biological security of beekeeping in the Mediterranean region. And thus, our aim is also to provide immunological power when using secondary metabolites directly against pathogens that contribute to colony collapse disorder, CCD. Voila, voila.
Thank you so much. We had a few problems with the internet. We managed. Thank you so much, Dalida. I really have lots of hopes on this work. And quickly go to Vasilis. Vasilis Stathopoulos.
Hello from me as well.
Just a few things about the precision apiculture tools.
Yes, we are within Safe AgroBee. We are two companies, Acme from Italy and Teraspatin from Greece. We provide precision apiculture tools for all the other partners. From our side, we have developed Scales, which is an IoT device, collecting measurements from the field, like temperature, humidity, weight, location, but also we combine them with space data in order to give an expression of the condition happens on the field. We work with LTE networks, but also with the narrow band. And what we collect, we present them on a web platform, which is under development. And for that platform, all the partners around Mediterranean participating in the consortium will have the opportunity to follow their apiaries without going all the time on the field. We aim to make it work more and more properly in order to fix any bug that is arised. And just a few images from the platform. This is the main dashboard where we have visualization of the places where we have put our devices. Here we have the total amount of kilos, and we can see also how many devices are active and how many have problems. We can see via GNSS the location of the devices and also to see if it is active or inactive to make zoom, unzoom, etc. What else we can see is the latest collected measurement in terms of humidity, temperature, and weight. At the same time, for these areas on the right side, we can see here the weather conditions which occur at these places right now, together with the collected, for example, environmental temperature. We can also see the battery, and we can provide the system with a photovoltaic panel or not, depending on the frequency of the measurements. There is another dashboard for the analytics, where we select the dates, we select the apiary, and we select the specific device, or we select the total devices of each apiary. And we have prepared a diadrastic, let's say, chart, where we can put everything together with different axes, and to see at the same time the weight, the temperature, the humidity temperature, and so on. We can export it in PNG, and we will make it to export it in CSV as well. We can select what we would like to present and what we didn't like to present in order to make a combination of the data. And we are now preparing another tab where we will put layers from Copernicus data for ozone, but also for the vegetation of the areas where we have put our devices. So in the same platform, we will also present the satellite data along with the data collected by the devices. And this tab as well present all the devices we have right now in Greece, but also in the other countries. As a summary, well, we can have an overview of what is active, what is inactive, what happens with weight, what happens with the elements, but also for the status of its device. So this was a short presentation of the platform, but also of the devices that we're developing as Teraspatium, while our partner, ACME, from Italy, is developing a sound, let's say, sound recorder for the hives. And they will be able to present it during the next, let's say, meeting. So this was an overview, and thank you very much for your attention. I try to be very short.
Very fast, very fast. Thank you so much. We must say that ACME 21, the sensors they are making for inside the colony, it is temperature, humidity, sound, and now they are preparing also an accelerometer. And for this type of devices that Vasilis showed you, in Safe Agro, we will have something about 10 or 15 per country, I forgot. But then we share similar type of balances with another project between that we have between France, Germany, and Greece. And in Greece, this type of devices, they are 45 in 9 different places. So in fact, it's quite a lot of data being collected from the 2 projects. So, okay, we finished with Safe AgroBee. Thank you all. And I would like to ask Emilio. Emilio, please, you've been so kind waiting for us. Please tell us about your project, the new one.
Okay, thank you very much. Yes, this is a project yet to start. We will have our kickoff meeting in 10 days. And basically, this is not a project on the bees, but I think that has a deep effect on the bees, particularly wild bees, because the project, as you can see from the title, is based on the development of agroecology protocol to enhance the resilience and the ecosystem services in tomato crops. So this is an outlook of the people involved. We are 3 partners from Italy, so we have partners from Portugal, from Spain, from Greece, from Turkey, from Algeria, from Morocco, Tunisia. So we are a very big consortium. This is the project at a glance. So you can see that basically everything is based on the protection and increase of biodiversity, both above ground and below ground. And the main aim is to reduce the chemical inputs in tomato crop. That is one of the largest crop in the Mediterranean basin. So by reducing the input with fertilizers, herbicides, and particularly pesticides, we hope to develop strategies that will increase or at least to halt the decrease of wild pollination in this system. When I say ecosystem services, because the other one, the other 2 services that we are focusing on are the fertilization, so how to improve the health of the plants and the nutrition of the plant without giving synthetic fertilizers, and most of all plant protection, because one of the main causes of bee and wild pollinators decline is the use of pesticides, and pesticides are essential to protect the crop, but now we have knowledge to reduce them in a way that is not affecting the final product. Actually, we can produce maybe less, but of higher quality. Quickly, an overlook of this is the the first work package that is focused on the biodiversity below ground. So what we are doing is just to characterize the soil microbiota that could help the plant either for nutrition and for protection. So we have people extracting from soil samples the main microorganisms that could help plant in nutrition, just to name one, rhizobia, or those fixing the nitrogen, or those contributing to like mycorrhizal fungi that help the plant, particularly in stressful environments like those in the north part of Africa. The second part, the third work package is the one focused on the above ground biodiversity, and here is the key place of bees and wild pollinators, because what we want to do is to understand how increasing, particularly with the companion plants, intercropping and border cropping, the presence of bees and natural enemies of insect pests in tomato crops. So this part, particularly for the bee part, is cured by Gennaro Di Prisco, that probably some of you know very well, and he has suggested protocols for preserving the wild pollinators and for collecting them. We are using Malaise traps and colored pan traps. To reduce the synthetic inputs, we are also concentrating our efforts in studying what could be used to protect the plant from pathogens and pests, and so we have scheduled a number of laboratory and field tests with essential oils that are considered more environmental safe in respect to synthetic insecticides. The same consideration applies to the elicitor of resistance. There are a number of compounds that elicit plant resistance response, so these goes into direction of reducing the synthetic inputs for plant protection. And then there is a task occurred by the Turkish group that is studying the importance of using the crop residues and in an optic of circular economy. So we are not throwing away what remains from the crop, but we are using them with specific treatments to give to the soil, again, to give back some components, reducing the input of fertilizer. So at the end of the project, our aim is to build up a big protocol for farmers to reduce drastically the synthetic inputs, and to have a product that is also from a qualitative point of view better than a conventional one. And indeed, there is a specific task here focusing on the features. That means the quantity of main components of tomato, like lycopene, beta-carotene, that are important antioxidant compounds, so improving the tomato quality. And what we are aiming to do is just to understand whether the reduction of insecticides, fertilizers, and herbicides contributes to the enhancement of these important components of harvest. There is also, an environmental and economic assessment, because whatever we do in agriculture has to be economically feasible. So we cannot propose to farmers to do something that decrease the crop and reduce the quality. So what we want to push is just to make farmers aware of those environmental costs that are never considered by farmers. So when you are spraying an insecticides and you have 10% more product, you are not considering the environmental costs. You are just considering what you have saved in terms of production in respect to the pest or the pathogen that have attacked it, and that's the end of the story. But indeed, there is also an environmental cost that is the deployment of the soil, the deployment of natural enemies in the culture, and these are all problems linked to the extensive crop cultivation that is the idea that has dominated the agriculture in the last decades. So this is the implementation, and at the end, of course, there is also a big work package on the uptake dissemination. We have set up already some didactic farms, and we are scheduling a number of courses, particularly directed to women in the northern part of Africa, because apparently there is a great movement of new women farmers that need to acquire this new concept of agriculture, and for the development not only of the country, but also of the women condition in these countries. I think so. These are the acronyms of all the participants to the project, divided into the different activities. As you can see, there is a great part shared among most of the partners, that is the field, because this is a field-based project. We have reduced the laboratory part to the minimum just to give farmers immediately some practical results of the strategies of agroecology approach for the protection, the nutrition, and the pollination of tomato crops. This is the interaction among the VPs. And I think that that's it.
Thank you so much, Emilio. You were really very fast.
Yes.
And very informative. Yeah, well, your project sorry?
No, I just want to add that to assess the impact of our strategies on the biodiversity, particularly above ground, we are collecting ex ante and ex post the activities of the project with malaise traps. So we are targeting the Hymenoptera. Hymenoptera is the bioindicator group that we have chosen because it groups together the main pollinators and the main natural enemies that are Hymenoptera parasitoids of insect pests. So Gennaro will take care of the identification of the wild pollinators, and in case any of the project that I have see today are ongoing on bees, I don't know whether some of you would be interested in having some samples from the fields that we are sampling. Gennaro Di Prisco will update you, and you just have him as the main contact as for pollinators in my project. Okay, thank you.
Thank you so much. In fact, what I find very, very interesting is that you are also looking into the effect of this chemical input in the health of the plants, together with the insect pollinators, and particularly I believe that the first one enemy for the bees is the agrochemicals, and of course then everything else comes together, but that's the first one. So we finished with the hard part, and now just to lighten you up, we have a very nice video by a beekeeper, because he wants to salute the celebration. I wish we had more time and more more discussion time also to exchange more ideas, but we can continue discussing if you wish, and more partners to be involved, because I can see several of my Safe AgroBee partners here. And I just got a message says that the bees, the queens for work package 2 of Safe AgroBee, Diana, are ready to be exchanged, at least for Greece. So yeah, well, we're continuing working during the Zoom.
داني جعجع من برقة، قضاء بعلبك الهرمل. أنا عامل هندسة مدنية، قيادة وتنفيذ أشغال. تخرجت بال 2003 واشتغلت ببيروت لحديت ال 2010. بال 2010 كان القرار المصيري اللي أخذته، أهم قرار أخذته بحياتي، هو إنه أخذت قرار أترك المدينة وأطلع اشتغل بالضيعة. بالضيعة ما فيك تشتغل بالهندسة، فبقى انتقلت على دومين الزراعة وتربية الحيوانات. فتحت مزرعة بقر وفت بزراعات عضوية. فبقى بال 2016 بلشت بالنحل. ما كان عندي ولا أي خبرة بالنحل، وإذ شوي شوي جبت 5 أفران بلشت أتعلم فيهم، وصرت أحضر دورات وأحضر على اليوتيوب كتير شغلات. فبقى صرت أتعلم شوي شوي. هلأ بال 2017 2018 صار في مؤتمر النحالين العرب يلي صار بجية ببيروت، رحت حضرته وشاركت فيه، وهي كانت نقطة التحول إذا بدكم بحياتي النحلية. الحلو بقطاع النحل انه بيعلمك كتير من المثابرة وطولة البال بشغلك، وبيعلمك التنظيم بحياتك، بيعلمك العمل الجماعي، إذ انه بتصير تتمنى الخير لغيرك اكثر ما بتتمناه لحالك، لانه الخير بقطاع النحل بالاخص اذا ما فاض من عند غيرك ما ممكن يوصل لعندك. في كتير صعوبات وتحديات بتواجه المزارع بشكل عام والنحال بشكل خاص. أهم هذه التحديات هي إذا بدك تحديات المناخية اللي عم نمرق فيها. بطبيعة الحال النحل بيزيد من الإنتاج الزراعي بشكل ملحوظ. هلا في اشياء الواحد ما بيشوفها بالعين المجردة، بس اللي عم بيتابع مزروعاته واللي عم بيتابع بستانه ومزرعته بيقدر يلاحظ هالشي بكل سهولة. إذ بتحسي إنه الإنتاج نوعيته تغيرت وكميته أيضا تغيرت. هلا العبرة اللي أنا قدرت أخذها من خلال تعاطيي مع النحل، واللي بحب شاركها لكل النحالين أو لكل المبتدئين واللي حابين يفوتوا على القطاع، هو إنه ما تيأسوا. ممكن تجرب أول مرة وما تزبط معك، وتجرب ثاني مرة وما تزبط معك، وتجرب ثالث مرة وما تزبط معك. أكيد المشكلة مش بالنحل، المشكلة فيك لأنه مش عم تعرف تشتغل. ما تخلي اليأس يفوت على قلبك، على طول طور حالك قدر الإمكان ورجع جرب، ولابد ما توصل. الحلو بالنحل إنه فيك تشتغلوا مع شغلك، مش إنه بده كل وقتك.
Well, I think that with this very beautiful message for a beekeeper and the concept that he introduced, it's nice to go towards the end of this meeting. So I think we still have some time, although we are late, for maybe questions and answers, or you can reach all the partners of all consortia easily, I suppose, or any remarks from anybody. Would you like to add something? Just feel free as a family. Danny, can we please unmute everybody? Can we do that? Any comments? Anybody who would like to say something? Or you are tired? I suppose what we all say, yes, please, Sarah.
Yes, well, this conversation and the presentations have been really interesting. We've looked at how much things are in common and how many topics, you know, are correlated, and how we can open up into new collaborations afterwards, which is quite interesting and very important for such an event to enhance these collaborations, which we as PRIMA very much encourage, and we are very happy to see that like in practical terms, which is perfect. Many thanks for the event.
Thank you so nice. It's kind of we can create a kind of a cluster, in fact, between PRIMA with projects that they have common interest. And exactly. I can tell you that there are other 2 PRIMA projects, the Med Food TT Hubs, yes, and MediFeed. Yeah. And these people on 13th of May, they gathered in Thessaloniki and invaded, literally invaded in my institute, and we had a very nice visit and a very nice overview of what we are doing. I took them around and spending with them. More onto the food and honey. So if we don't have any other remarks from anybody or any questions, yeah, maybe we can have a common conference, in fact, all PRIMA projects later on. I don't know when. Just I'm really open to suggestions. We would like to finish this. Yes, Danny. You are muted. You are muted still. You are talking.
I was saying that I'm familiar with side events. You know, whenever there are big events, some people would go to some side events. So maybe we can take advantage from Apimondia or any other big event. We can be gathered in a booth or just have 2-3 hours together in one of the rooms.
That's a very good idea. And in fact, before we finish with a very nice video by FAO for the WBD Day, we should invite you to attend the Apimondia Congress in Istanbul in August. I don't know if you have already submitted abstracts, but if not, you can attend the other people that they're going to speak or present their work, or we can have this very nice of meeting. I really like the idea. So, all right, then we are already late, 20 minutes late. We can show you for half a minute the last video by FAO, number 1, and then we can say goodbye. Well, that's it then. A big thanks for everybody for your time, for your energy, for the willingness to participate. And yeah, I think that it was one of my best celebrations for the WBD Day.
Thank you. Bye.
Thank you,
Have
everyone.
a nice afternoon and be in touch.
Bye-bye.
Bye-bye.
Bye-bye, everyone.
Bye.